8.
Thank you for the induction.
Damn, he sucks.
Yeah. Fuck. Sorry, it's not natural. It's not.
Take 2. Ready?
Oh my gosh, remember Mr. Urbana?
Oh hell no, bro.
Wait,
Maybe I'll just be like,
you
let me
handled
just-
that so okay. Lucas, you are in front of an auditorium.
Let me. Shh. All right. Thank you for the introduction. I'm Lucas Hoffman. Thank you for coming out to the seminar today. I will be giving a presentation on how sugars and other polyols affect the chlorination rate of phenolic compounds. So first, I want to give some background into the water systems that this is applicable to. There's many municipal or wastewater systems that are treated with chlorine, and the intent of treating these systems with chlorine is to inactivate pathogens. But in this chlorination process, the chlorine can also react with natural organic matter in the water, and this can form disinfection byproducts. And you can see that from the diagram here. So what are these disinfection byproducts? These are harmful molecules that are cytotoxic, so they affect the health of cells. They're carcinogenic, so they can increase the chance of cancer. They are genotoxic, so they can alter genes, and known endocrine disruptors, so they can affect the transport of hormones. And it's potential. There's a lot of research that goes into the toxicity of these molecules. And the disinfection byproduct is generally more toxic than its parent compound before becoming chlorinated. So I want to talk about the organic precursors that we are talking about in this research. And we are specifically looking into phenolic structures like ethylparaben at the top there and phenol at the bottom. And these structures are denoted by a phenyl with a hydroxyl group coming off of that. And that is what makes it the phenolic structure. As you can see, phenols at the bottom, it's just phenol. And the phenolic structure is coming off of ethylparaben on the left side. And we looked into these precursors because these precursors are known disinfection byproducts, and they also have reactivity towards becoming chlorinated with electrophilic aromatic substitution. And electrophilic aromatic substitution is how these molecules undergo chlorination, and I have the mechanism at the bottom here. The precursor phenol here is acting as the nucleophile and is attacking the electrophile. And the hydroxyl group in this reaction, as you can see in these intermediate brackets, is forming a stabilizing electron donating interaction that decreases the, that makes the reaction go by faster. I also want to talk about a study that influenced the study I'm talking about today. So previous members of my lab group did a study on how methanol affected chlorination rate of phenolic compounds. So on this graph, I have chlorination rate as a function of methanol concentration. And as you increase the methanol concentration, the chlorination rate of ethylparaben and ethyl-3 hydroxybenzoate increases. So this is showing that the polyol concentration is acting as a rate enhancement, not the polyol, sorry, the methanol is acting as a rate enhancement for the chlorination of these phenolic structures. And as you can see, methanol is over there, and it has one hydroxyl group. And this study postulated that it was this hydroxyl group was stabilizing the activated complex during electrophilic aromatic substitution, and that is what caused the methanol volume to increase the chlorination rate. So that brings us to polyols. Polyols, unlike methanol, have several hydroxyl groups. And by definition, polyols must have 2 or more hydroxyl groups. But unlike methanol, there are several different characteristics that polyols can have. They can have several carbon chain lengths, they can have several hydroxy groups, they can have several positions of the hydroxyl groups, and also they can be cyclic, non-cyclic, or a combination of the 2. And here on the right, we have several different structures here. So you can see polyols, they're very different, they're very unique. Unlike methanol, There's so many different possibilities with these polyols. So it is interesting to see how these polyols could affect the chlorination rate of these phenolic compounds like methanol did. And why would this be important? Why would this research be important? It's because there are several anthropogenic processes that lead to these polyols entering water systems, such as food or beverage production, alcoholic beverage production, fruit or sugarcane processing, paper or pulp processing, and even urine, and especially urine of diabetic individuals where the glucose concentration is high. So there's a lot of these processes where polyols are entering waters that could possibly become chlorinated. So the purpose of this research is to determine how several phenolic structures are affected by, the chlorination rate of phenolic structures are affected by polyol, specifically polyol concentrations. And as you see here, we have several different polyols: glucose, maltose, sucrose. So now into my methods. So the independent variable of this research was polyol concentration, and this affected the dependent variable of the chlorination rate. We also looked into several additional factors on top of the independent variable, such as the polyol identity, the precursor identity, temperature, pH, and NaCl concentration. So there are several factors that we looked into to kind of get more information of the systems that these polyols will be in and be more applicable to systems that are becoming chlorinated, such as trying to match water treatment facility pHs or concentrations or specific precursors that are prone to becoming chlorinated. This was conducted with time course experiments. And if you're not familiar with time course experiments, I'll go through a quick diagram real quick. First, we start with a reactor, and in the reactor we have a set buffered pH, we have a set salt identity controlling for ionic strength, and then we added a polyol with specific concentrations, so that would be glucose, maltose, sucrose, and then we added free available chlorine, which in all cases was hypochloric, or most cases was hypochloric acid, and this was in all cases around 300 micromolar to 200 micromolar. And lastly, in the reactor, we would add a nucleophilic aromatic precursor such as phenol or ethyl paraben, and once we spike in this precursor, it would begin the reaction. And once the reaction begins, we would take samples every 20 or so seconds, but it depends on the rate of the reaction. If it's faster, sampling faster. If it's a longer reaction, the longest I did was about maybe 12 minutes, so it was long between samplings. It was like 2 minutes between each sample. So it really depends. But these samples are, these aliquots from the reactor are put into HPLC vials, and they're pre-amended with sodium thiosulfate, which is a quenching agent, and this essentially stops the reaction at specific time points. And these samples were then run on HPLC alongside an external calibration standard set, and this would then give a mass balance. And this mass balance is concentration as a function of time. But since this data is collected in a reactor that was set at pseudo first order, we are able to graph, sorry, I didn't introduce the reaction. This reaction is 4-chlorophenol becoming chlorinated to 2,4-dichlorophenol and 2,4,6-trichlorophenol. But that's not as important right now, but just showing the reaction that's occurring here. The parent compound is 4-chlorophenol, and that's what we're looking for, the decay, to show the reaction rate. But since this isn't linear, it's kind of hard to extract data from it. So we can linearize it once, as I said earlier, because it's pseudo first order reaction, we can plot ln of the concentration of 4-chlorophenol as a function of time. And that gives us a linear line that the negative slope of is the chlorination rate. And that's how we determine the chlorination rate from all the different reactors we ran. All right, so now I'd like to get into findings. The first condition we looked into was polyol identity. And this graph here is showing chlorination rate as a function of polyol concentration. And the slope here can be seen as rate enhancement. So the more sensitive the slope is, the greater the rate enhancement. And I have 3 sugars on this graph: sucrose, maltose, and glucose. Sucrose has the most sensitivity towards polyol concentration in the chlorination rate. I also forgot to mention that this is of the chlorination of ethyl paraben. But yeah, as I was saying, sucrose was the most sensitive to this rate enhancement. It caused the greatest rate enhancement for the chlorination of ethyl paraben. So it was used for most future experiments. Next, I would like to talk about polyol carbon chain length and why it matters. And this kind of goes hand in hand with the polyol identity, as this is one of the characteristics of the polyol. This graph here is the chlorination rate of a reactor with positive polyol over the chlorination rate of a reactor with negative polyol. And that just means the negative polyol is always going to be 0 millimolar concentration of polyol, while the positive is either going to be 5 millimolar polyol if it is the blue bar, and 50 millimolar polyol over for the red bar. And that is essentially, the y-axis is just rate enhancement. And that is a function of carbon chain length. And we tested several different polyols of several different carbon chain lengths. And we found that that there's a major fluctuation between 3 carbon chain and 4 carbon chain. And then after that, once you go to 5 to 6 to 12, there's more of a linear increase of the rate enhancement as you increase the carbon chain length. And yeah, that's basically what I'm just saying at the bottom there. Next, I want to talk about the number of hydroxyl groups, as this is another characteristic of these polyols. This graph is the same, the y-axis is the same as the previous graph. It's still rate enhancement, it's still same parameters, but now it is a function of hydroxyl groups. And 2 hydroxyl groups, you can see that there's a huge rate enhancement for the 50 millimolar polyol over 0 millimolar polyol. But after that, it drops down to 3 hydroxyl groups, and then there's more of a linear increase, such as we saw in the carbon chain length, from 3 hydroxyl groups to 6 hydroxyl groups. And using all this data together, the polyol identity, the carbon chain length, and the amount of hydroxyl groups, it's important to take them all together. And the findings that I found from this is that there's 2 factors that really matter on how the polyol enhances the chlorination rate of these phenolic compounds. And that is the availability of hydroxyl groups and the steric effect of the hydroxyl groups. It seems as if there's a steric effect of these polyols and they're not able to get as close and be able to stabilize these phenolic structures, then there's not as much rate enhancement. But as soon as you get a lot of hydroxyl groups on that polyol, just due to the sheer availability of hydroxyl groups, the steric effect will be overcome and there will be an increase in rate enhancement. That's what we can see from the previous graphs and this graph right here. Next, I would like to talk about precursor identities. That's the next factor we looked into. This graph right here is the chlorination rate as a function of sucrose concentration for phenol, and a lot of its chlorinated, all of its chlorinated derivatives, as you can see on the right there. A takeaway from this graph is that there's differences in rate enhancement for isomeric compounds. So even though 2CP and 4CP, over here, this is 4CP, that's 2CP, they have the same molecular formula. Excuse me, they have the same molecular formula. However, their rate enhancement is drastically different. And this is because there's isomeric differences. It matters about where the position of the chlorine is, and it matters about the structure of the phenolic compound. Since the chlorine is on the ortho position, it increases the reactivity. And that can also be seen with 2,6. Dichlorophenol, all the way on the middle here, is 2 ortho positions are occupied, and that increases the rate. So when these ortho positions are occupied, it increases the rate enhancement of sucrose on the chlorination rate of these phenolic compounds. So yeah, isomers are influenced differently, as I was explaining. Next, we looked into boctyrosine. And boctyrosine is interesting because it's an amino acid. And since it's an amino acid, it's applicable in biological systems and especially proteins. So that's a very interesting, that's why we looked into boctyrosine. Other than it being boctyrosine because it has a blocked amide group, so there's no side chlorination reactions other than it reacting with the phenolic structure. But the same trend can be seen with this boctyrosine and its chlorinated derivative as as the ortho position, if it's occupied by the chlorine, it increases the reactivity by double here. The slope doubles from 1.46 to 2.8. So it's interesting to see that the chlorine occupying the ortho position increases the rate enhancement of these phenolic structures in the presence of sucrose. Next, I would like to talk about the thermodynamic activation parameters. And this was found through several time course experiments where temperature was changed. So first we have a graph here that is chlorination rate as a function of temperature. And this is for the chlorination of phenol at 30 millimolar concentration sucrose. This graph is parabolic, so it's not linear, there's not much data we can extract from it. But if we use the Irene equation down here, this would be the y-axis and this would be the x-axis excluding the enthalpy. We can get ln, chlorination rate over temperature, as a function of 1,000 over temperature. And this gives a linear line that we can use the y-axis, we can use the x-axis, and the slope to determine the enthalpy and the entropy of the activated complex, which we found down there. And then this was conducted for several different sucrose concentrations, and then a graph was compiled of enthalpy of the activated complex as a function of sucrose concentration, and also for entropy of the activated complex as sucrose concentration. And the activated complex is, in electrophilic aromatic substitution, is aromaticity breaking. So this gives insight into how polyols are affecting, specifically sucrose, is affecting the activated complex during electrophilic aromatic substitution. So for both of these graphs, we have enthalpy and entropy of the activated complex decreasing as sucrose concentration increases. And this means for enthalpy that the activated complex during electrophilic aromatic substitution, the energy is decreasing due to sucrose increasing. But for entropy, that means that as the sucrose concentration is increasing, the structured lattice of polyols around the activated complex is increasing. So it's becoming more structured as you increase the polyol concentration. And those are the important takeaways from the activation parameters and how they were found. Next, I would like to talk about the influence of pH on the rate enhancement of these phenolic compounds in the presence of sucrose. This graph is showing the chlorination rate as a function of pH for 50 millimolar sucrose at the top and 0 millimolar sucrose at the bottom. And it looks like from this graph that there is an effect of pH on the rate enhancement. However, there is actually not, because the factor of rate enhancement from 50 to 0 millimolar is a factor of 4 regardless of the pH. So there's no difference in pH, and the rate enhancement is independent. Next, since there was no effect on pH and pH was independent of the rate enhancement, we conducted experiments switching the pH and NaCl concentration of the reactors. So this graph is the ratio of chlorination rate of 50 millimolar sucrose reactor over the chlorination rate of a 0 millimolar sucrose reactor as a function of pH. With 3 different data sets here, with 5 millimolar NaCl, 30 millimolar NaCl, and 60 millimolar NaCl. And what was seen, there was trends that as you increase the NaCl concentration of the reactor while also decreasing the pH, this would cause the rate enhancement to decrease closer to 1. And this makes sense when you look at the reaction equilibrium once you're adding NaCl. We have hypochloric acid as the chlorinating agent in all the reactions. There's the same concentration as I said in the beginning. It was around 200, 300 micromolar of hypochloric acid as the chlorinating agent. But since we're adding NaCl now, from this equilibria, you can see as you increase the Cl concentration and you increase H+, so you make the reactor more acidic, it pushes the forward reaction towards the formation of Cl2, which is another chlorinating species. And this switch in chlorinating species seems to affect the rate enhancement, and it actually decreases the rate enhancement of these polyols as the chlorinating species switches from hypochloric acid to to Cl2. And, oh, sorry. And this is interesting because that means hypochloric acid is very important as a chlorinating species to cause the rate enhancement of polyols in the chlorination rate of these phenolic structures. Lastly, for findings, I would like to talk about the postulated mechanism. So this is kind of bringing it all together of what is happening. How are these polyols stabilizing these phenolic structures during electrophilic aromatic substitution? What we have here is the postulated mechanism. So at the top is the structure of sucrose, and at the bottom is a phenolic structure undergoing chlorination. And the hydroxyl group of sucrose is stabilizing the phenolic structure during chlorination. And this is happening through the hydroxyl group forming hydrogen bonding with the hydroxyl group on the phenolic structure, and through forming stabilizing interactions with the ortho hydrogen through secondary electrostatic interactions. So overall, the polyols are stabilizing the reaction occurring here, and that's how they're causing the rate enhancement. So why does this matter? Why is this important? Why should you care about this research? Well, how many people here are coffee drinkers? Raise your hand, I know you're a coffee drinker. So this research, see, that right there. So this research matters to you then, because in coffee, let's say you put some sugar in it, some table sugar, and that would be sucrose. And then let's say you put tap on it, or any water. Most water is going to have residual chlorine in it. Most waters are going to have residual chlorine. And then, lastly, you have the coffee beans that have the phenolic structures in them, or the nucleophilic aromatic precursors that we were talking about. It could be phenol, it could be phenolic structures, or it could be other compounds that we haven't even looked into the study that could be rate enhanced by polyols. But you have all of these reactants in your coffee, so what I just talked about polyols enhancing the rates, they could be enhancing the rates of disinfection byproduct formation in your coffee, and you could be consuming more of these harmful molecules. And obviously that's not good for public health. But yeah, it's also applicable in the systems I talked about earlier, the wastewater and municipal systems that are treated with chlorine. There could be residual polyols in these waters, and they could be reacting with natural organic material to increase the formation and possibly even the yields of these disinfection byproducts. And also processes such as bleaching paper, that's also in bleaching of other sources, possibly nuts, anything that can have disaccharides or monosaccharides like the ones we talked about in this study. So this leads back to disinfection byproducts, which are not good for public health. So these disinfectant byproducts that are forming from increased polyol concentrations could be formed at a faster rate due to the presence of these polyols. And these disinfection byproducts are more ecologically and toxicologically hazardous than before they were chlorinated. I meant to take that out. That's not supposed to be it. Honestly, this slide in total, I probably don't need it. But lastly, I wanted to talk about in-body biological systems, because this is a really interesting applicability of this research that I hope to pursue more of. But as I explained earlier, how bactyrosine can become chlorinated, and it was even seen that there was a rate enhancement in the presence of sucrose for the chlorination of this amino acid. That can mean that other amino acids as well could become chlorinated, and that could lead to oxidative stress, especially at sites of inflammation in mammals. And that's because there's actually an MPO enzyme in our body that produces hypochloric acid, the same chlorinating species that I used in this research, the same chlorinating species that is used in wastewater and municipal water treatment. It's being formed in your body, probably as we speak right now. And it's interesting to think about how our sugar concentration in our blood is actually affecting the formation of these disinfection byproducts, or not disinfection byproducts, but oxidative stress within the body, chlorinating these amino acids and all these proteins that you probably don't want to become chlorinated. Lastly, I would like to acknowledge Ronald and Linda Raspet for providing me a stipend to be able to research during the summer, and U.S. National Science Foundation and the Camille and Henry Dreyfus Foundation for supporting the paper that I co-authored. And lastly, I want to thank my lab group and Dr. City as my PI. Thank you.
I love your transitioning.
Dude,
No,
no,
I was writing it
that-
down.
And then when you had those moments.
Oh, and it was 25. That's perfect.